In your first semester, we have studied under the paper that is biochemistry some of the topics like carbohydrates, fats, amino acids, proteins and nucleic acids. In this semester, we will continue it, but only one particular class of molecule, that is nucleic acids. What is the structure of nucleic acids? What is their function? How the function is related to their structure? All this we will be studying in this semester in this paper. This paper, designated as molecular biology, will contain all this information, and which is very thrilling and fascinating. We will be finishing in this term. Many of you must have seen a Hollywood movie released few years back where the recreation of dinosaurs was shown. From a fossil of age old, an insect was isolated from which a chemical was isolated. From this chemical, the huge animals, the dinosaurs, which were extinct, were created back. What is this chemical substance? How is that it helped in creating the extinct animals? Where it resides in a cell? The answer is DNA, a three-letter abbreviation for the name deoxyribonucleic acid, the hereditary material. By about the middle of this century, the role of chromosomes in inheritance was well established. Again, the question is, what are they made of? The analysis revealed that they were composed of protein and an acid called DNA, or deoxyribonucleic acid. This substance of genes is the most celebrated and fascinating chemical of 20th century, continuing its uniqueness even in this 21st century. The term biotechnology is today centered around this material, revolutionizing the modern biology. DNA was first isolated in 1869 by Friedrich Miescher, a German chemist, and so named it because they are found in the nuclei of pus cells. This substance was largely ignored by biologists for nearly a century after this discovery. The reasons were it seemed far too simple and uniform in its structure to serve a very significant purpose. Today we know that this macromolecule is the genetic material. The reasons were it seemed far too simple and uniform in its structure to serve a very significant purpose. Today we know that this macromolecule is the genetic material. Chemically speaking, your genetic endowment consists of DNA inherited from your mother and father. Of all the chemicals in nature, DNA is unique in its ability to direct its own replication or synthesis. The resemblance of offspring to their parents has its molecular basis in the precise replication of and transmission of DNA from one generation to the next. The modern biotechnology advanced so much that today it is possible to make or alter the DNA in the laboratory and insert it into a cell, changing the cell's heritable characters to the advantage of mankind. It was known by 1940 that chromosomes of the cells carry the hereditary information containing both proteins and DNA. In the beginning, most researchers considered protein as the material of genes, since little was known about nucleic acids at that time. However, this view gradually changed as the experiments with microorganisms yielded unexpected results supporting DNA as the genetic material. Before we go into the details of the structure of DNA, we will briefly see the experiments favoring and confirming the DNA as the hereditary material in living systems. Frederick Griffith A British medical microbiologist was the first one to present evidence in favor of DNA as the genetic material in 1928. He was working with Streptococcus pneumonia, a bacterium causing pneumonia in mammals. He found two different strains of this bacterium, one producing colonies that appeared smooth and the other strain appearing rough. Cells of the smooth strain, designated as S, were surrounded by a polysaccharide coat or capsule which was missing in the rough cells, designated as R. When Griffith injected the bacteria into mice, he observed that only the S strain was pathogenic and not R strain. Mice injected with S cells died of pneumonia and those with R cells survived. However, when S cells were heat killed, they were found harmless like R cells to the mice. Following this, he mixed heat killed S cells, which are non-pathogenic, with the live R cells and injected the mixture into mice. Although none of these two were pathogenic, individually mice injected with the mixture developed pneumonia and died. Moreover, Griffith found live S cells in the blood samples of dead mice, in spite of the fact that only the dead S cells were injected. Somehow, some of the R cells had acquired from the dead S cells the ability to synthesize polysaccharide coats as an heritable character, getting themselves transformed into live S cells. When S cells taken from dead mice on cell division produced daughter cells with coats. This phenomenon, discovered by Griffith, is now known as transformation, that is the assimilation of external genetic material by a cell. Although the chemical nature of the transforming principle was not known by that time, Griffith's observations spurred other scientists to search intensively for the elusive genetic material. Additionally, his experiments with heat-killed bacteria also hinted scientists that the genetic material may not be proteins as they get denatured on heating, yet the S cell genetic material remained able to function in transformation. Following Griffith, Oswald Avery, an American bacteriologist, pursued this work for a decade to identify this transforming principle by purifying various chemicals from heat-killed S cells and then testing each substance with live R cells to examine its ability to transform the bacteria. Finally, in 1944, Avery and his colleagues, Maclean McCarty and Colin McLeod, announced that the transforming agent had to be DNA, as its properties resembled that of DNA rather than protein. At that time, the information about DNA was so little that many biologists could not be convinced with the discovery that DNA was the genetic material. Avery's experiments to tackle the problem is presented here.